Hello everyone. Today we'll be solving Cambridge International AS and A Level Biology Paper 4 Structured Question, February-March 2021, Paper 42, Part 2. Question 6. Dogs have exceptionally good sense of smell. They can detect molecules in the air with a concentration of 1 parts per trillion. Olfactory receptors are the sensory receptors in the nasal cavity that respond to chemicals in the air. They function in a similar way to the chemoreceptors of cells in the taste buds of the tongue. Describe how olfactory receptors in cells in the nasal cavity of dogs respond to chemicals to generate an action potential. So basically, chemicals stimulate the receptor cells, and as a result, sodium ions enters the receptor cells via the microvilli. These increases positive charge in the receptor. So this increase in positive charge in the receptor is the receptor potential, and this causes the membrane to be depolarized. As the sodium ion enters, the membrane gets depolarized. If the depolarization reaches a certain threshold, like minus 55 millivolt, then voltage gated calcium ion channels open, and calcium ion enters the cell. The vesicles of neurotransmitter move towards the cell surface membrane, and the neurotransmitter is released by exocytosis. The neurotransmitter then binds to receptors. The receptor cell acts as a transducer. Part B. Dogs can use their sense of smell to detect differences between breath of people with lung cancer and breath of people without lung cancer. These differences are due to the presence of particular chemicals at a very low concentration in the breath of people with lung cancer. Dogs can be trained to sit when a person's breath indicates that lung cancer is present. Screening people for lung cancer is important for early detection and treatment. Suggest two advantages of using dogs in the way to screen people for lung cancer. Well, using dogs is a very non-invasive process. It is very cheap, all right, and this allows early detection of lung cancer. This procedure can be done at home. Question number 7. A. Figure 7.1 shows two chromosomes in one stage of meiosis. The letters G to M represent the dominant alleles for 7 genes, and the letters G to M represents the recessive allele of the same genes. Okay. Name the structure labeled R and S on figure 7.1. So we can see R are sister chromatids. And S is centromere. Part 2. State 3 features visible in 7.1 that identify the chromosomes as homologous pair. So we can see that they are this homologous pair because they contain the same genes. First thing first. And then these genes are located at the same location. So same loci. They have the same position of the centromere. As you can see, they are in the same position of the centromere, and they have the same length. Part 3. Figure 7.2 shows the same two chromosomes at a little later in the same stage of meiosis. Crossing over is beginning to occur at point T. So we can see that a crossing over at point T is going to occur. Okay. Now, figure 7.3 shows an outline of the same two chromosomes after crossing over has occurred. Complete figure 7.3 by writing in the letters of the alleles along both chromosomes. Take care to clearly show the differences between letters representing dominant alleles and letters representing recessive alleles. Okay, so we can see crossing over has occurred at this particular point. So we're going to mention G, H, I, J, J in the same location, K, K, L, and they are going to be in the same location. Smaller k, lowercase letter, they are not exchanged, so they're going to be in the same location as well. J is not exchanged, so in the same location. I, H, and G in the same, and G in the same location. However, so capital G, capital H, and I will be present in this chromatid, and small g, small h, and small I will be present in this chromatid. State the stage in meiosis in which crossing over occurs. So crossing over occurs in the prophase 1. So prophase 1, writing that 1 is very important because it occurs in the prophase 1. C, crossing over results in genetic variation. Explain how random assortment of homologous chromosome also results in genetic variation. Homologous chromosome, they can align independently of each other at the metaphase plate. This leads to different combinations of chromosomes in the daughter cells. This also results in a new combination of alleles. In humans, 2 to the power of 23 different combinations are possible. Question number 8. 8a, figure 8.1 shows a transmission electron micrograph of a mitochondria. So we can see B being labeled is actually cristae. On figure 8.1, use the letter A with a label line to show a location where Krebs cycle occurs. We know Krebs cycle occurs in the matrix, so we can label the matrix A. And B is cristae. Part 3. The inner mitochondrial membrane is the site of oxidative phosphorylation. Explain how the structure of inner mitochondrial membrane is linked to its function. The inner mitochondrial membrane, we can see there is a lot of infoldings in the inner mitochondrial membrane. It produces a lot of cristae, which increases the surface area for the electron transport chain, ATP synthase, all of those proteins to sit in that particular membrane. The inner membrane is impermeable to hydrogen ion. So when hydrogen ion builds up in between this intermembrane space, the hydrogen ion is unable to directly migrate into the matrix. This allows the formation of hydrogen ion gradient or proton gradient. The inner membrane also consists of proton pumps. Part B. The activity of succinate dehydrogenase, an enzyme in the Krebs cycle, is used as a measure of the rate of respiration in the mitochondria. The redox reaction catalyzed by succinate dehydrogenase is shown in figure 8.2. So we can see succinate, it leads to fumarate. Succinate dehydrogenase gives off an electron. So that electron can reduce something. The activity of succinate dehydrogenase can be measured using redox indicator. Name a redox indicator. Explain why this indicator can be used to measure the activity of succinate dehydrogenase. So, DCPIP is a redox indicator. We know that DCPIP, oxidized DCPIP can be used for this particular process. Oxidized DCPIP has a color of blue. So it's, when the electron is accepted by oxidized DCPIP, it's going to go from blue to colorless. Part C. Figure 8.3 shows a ground squirrel, Ictidomis tridecemlineatus. In the winter, the ground squirrel curls into a spherical shape underground and sleeps for long periods. During this time, the ground squirrel switches between two states. One is the state of torpor, when the body temperature is maintained at 10 degrees Celsius. It's a mammal. It's supposed to maintain its body temperature at 37 degrees Celsius, but the torpor is like a hybrid condition. All right, okay. Then it goes to a particular state called euthermia, when the body temperature is maintained at 37 degrees Celsius. Scientists use the activity of succinate dehydrogenase to investigate the rate of respiration in the mitochondria of ground squirrel. Mitochondria were extracted from the liver and muscles and samples of ground squirrel. The rate of respiration was measured at different concentrations of succinate and at temperature that corresponds to torpor, 10 degrees Celsius, and euthermia at 37 degrees Celsius. The results are shown in figure 8.4, rate of respiration in arbitrary units. We can see as the concentration of succinate increases, rate of respiration at 10 degrees Celsius also increases, also at 37 degrees Celsius also increases. However, one thing that is for sure is that respiration in muscle cells is always higher, in 10 degrees Celsius and also in 37 degrees Celsius. Respiration of liver cells in both temperature is lower. Now let's move on to the next page. Question number 1, describe the trends shown in 8.4. So we can see in this figure 8.4, the rate of transpiration increases, the rate of respiration increases as the concentration of succinate increases. But this respiration increases only up to 1 millimole per dm cube. Any increase in succinate after that does not affect the rate of respiration anymore. The rate of respiration at 37 degrees Celsius, we can see, is higher in both, all right, is higher in both liver and muscle cells. The rate of respiration is higher in muscle cells, all right, in both temperature. At 37 degrees Celsius, at 37 degrees Celsius, muscles has 210 arbitrary unit of respiration. And at 10 degrees Celsius, the same muscle has 30 arbitrary unit of respiration. Part 2. Explain the difference in the rates of respiration between liver at 37 degrees Celsius and muscle at 37 degrees Celsius. You see, we can see that, muscles at 37 degrees Celsius has a very high rate of respiration compared to liver. That can be explained that muscles have higher rate of respiration because it needs to produce more ATP. It is more metabolically active, so it has a higher need for ATPs. Part 3. During torpor in ground squirrel, muscle tissues uses more energy than liver tissue. Suggest one reason for this difference. Muscle contraction generates heat, or generally we can say muscles are more metabolically active. Section B. Question number 9A. Compare the characteristic features of domain eukarya and bacteria. So when we want to compare the characteristic features of domains eukarya and bacteria, eukarya have nucleus, they have linear DNA, they have histone proteins associated with their DNA, they have membrane-bound organelles, they have ATS ribosomes, they have cell that divides by mitosis, they have multicellular cells. All right, they are multicellular. The cell walls are sometimes present. If the cell walls are present, they're either made out of chitin or they're made out of cellulose. Whereas in the case of bacteria, we can say that they do not have a nucleus, they have circular DNA. No histone protein is associated with their DNA. They do not have any membrane-bound organelles. They have 70S ribosome. Their cell divides by binary fission. They're unicellular. The cell wall is always present, and the cell wall is made out of peptidoglycan. Question 9b. Describe the methods used to conserve endangered plant species. Well, endangered plant species can be kept in botanic gardens. Research can be carried out on them, and controlled growing conditions can be provided in the, in a glass house. An example, light and water and temperature can be maintained. Number of plants can be increased by tissue culture, and then it can be planted back in natural environment. Endangered plant seeds can be stored in seed banks at very low temperature. The seeds can be regularly checked for viability to maintain genetic diversity. The seed can also be germinated before introducing back into the natural habitat. For question number 10A, explain what is meant by bioinformatics and outline the role of bioinformatics allowing the sequence of genomes of humans and parasites. Basically, bioinformatics is the analysis of biological data using computer software. The database contains vast information about DNA and amino acid sequences. The large database allows information to be shared fast and efficiently. Scientists can predict protein structure from the DNA sequence data. Scientists can use BLAST as an analytical tool for comparison. Bioinformatics can be used to find methods to control parasites by developing vaccines. Question 10b. Explain how microarray can be used to analyze gene expression in a tissue sample. So microarray contains probes. Probes in the microarray are single-stranded DNA. Each probe is unique to a particular gene. All the probes in microarray corresponds to thousands of genes. The mRNA from the tissue sample is extracted, and the mRNA is used as a template to make the cDNA out of it by using reverse transcriptase enzyme. The cDNA is then linked to fluorescent dye. The cDNA is then added to microarray. The cDNA binds to the probes by complementary base pairing. Excess cDNA is then washed off. The microarray is exposed to UV light. Fluorescence shows the expressed genes. The intensity of fluorescence shows level of gene expression. Guys, we are done with the question paper. Thank you for joining the class. We will be solving question papers like this every day, and we'll be uploading it in our channel. Please subscribe to our channel, and you then in that way you are going to get notifications when new videos are uploaded. And if you have any question paper of choice that you want us to solve, all right, let us know in the comments. We will be solving and uploading question papers every day. However, due to the intensity of the work and everything, you can let us know which paper you want us to solve, and we will queue it up in our next videos and we will upload it. Only chemistry and biology question papers will be answered in this particular channel. Guys, thank you for joining the class. See you in the next class.